Noir Plasma Chrome Bleed. Unlocking knowledge, inspiring innovation, empowering curious minds.
This website makes learning so easy. I found all my resources here.
Like what you share.
Sterile injury triggers complex molecular cascades. Necrotaxis drives immune cells toward damage while pathways dictate the inflammatory response. Necrotaxis drives sterile inflammation. Cell death pathways release cellular damps. Macrophages regulate iron homeostasis. Altered self-recognition triggers the cascade. The demise of a cell initiates a sophisticated cascade. With necrotaxis, living cytic cells migrate up chemical gradients. They target the precise coordinates of injury. The consequences are dictated by the specific cell death pathway. This report, we examine the regulation of iron recycling and the checkpoints governing immunological self-recognition. When a cell dies, it releases chemical beacons. These molecules recruit myeloid cells to the site. This process is called necrotaxis. Mitochondrial formulated peptides initiate necrotaxis. Upon injury, physical destruction spills them out. This release guides immune cells to the site. Peptides bind to FPR1 and FPR2. These are GPCRs coupled to inhibitory G proteins. They feature distinct pockets but share a conserved activation chamber. Upon agonist binding, G1 signaling triggers PLC. This promotes migration and generates harmful RSS. ATP spilled during rupture acts as a primary find-me signal. It is recognized by the purinergic receptor-induced rapid desensitization. This results in their complete inactivation. Subsequent stimulation of P2Y2 by extracellular ATP reactivates these desensitized, ligand-bound allowing them to resume downstream signaling and superupside production. In homeostasis, HMGB1 is a nuclear DNA chaperone, but upon traumatic cell death, it is passively released. Outside the cell, it acts as a critical necrotactic mediator.
Under stress, HMGB1 translocates. Then, the inflammasome triggers transport. It binds to TLR4. This initiates pro-inflammatory signaling.
Uric acid is released from damaged cells. Transporters move it into target cells. This active B pathway. Chemokines recruit macrophages. Crystals stimulate gouty inflammation.
Mitochondrial formulated peptides, or MTFPs, target FPR1 and FPR2 flux, driving directed myeloid cell migration and ischemia reperfusion injury.
ATP acts as a potent damp. It targets the P2Y2 receptor. This promotes phosphatase-sensitive reactivation of desensitized FPRs. This amplifies leucite migration.
HMGB1 is a critical damp. It targets TLR2, TLR4, and RAGE. This activates NF-kappa B and primes driving the transition to sterile inflammation.
Uric acid acts as a powerful damp. It targets urate transporters and TLRs. This activates the NF-kappa B pathway, driving transcription. This leads to macrophage. These are key annexin A1 and lipoxin A4 target FPR2 receptor. This promotes receptor homodimerization, suppressing PMN infiltration and enhancing wound healing.
The immunological consequences of cellular clearance are fundamentally determined by the kinetics, morphological changes, and molecular signaling pathways defining how a cell dies. Apoptosis is programmed cell death. The cell shrinks and chromatin condenses. Violent membrane blebbing then occurs. Apoptotic bodies form safely. Phagocytes engulf them in a clean process. Apoptotic cells must be cleared rapidly, otherwise they proceed to secondary necrosis. This occurs when phagocytic capacity is overwhelmed. The plasma membrane disintegrates. This leads to the passive leakage of damps.
Necrosis is an unregulated response. It is characterized by cell swelling and membrane rupture. This releases intracellular components. This triggers rapid leukocyte recruitment. When caspase 8 is inhibited, RIPK3 phosphorylates MLKL, driving its translocation to the membrane. Here, it creates physical pores, causing osmotic swelling and lytic burst.
In healthy cells, flipases maintain membrane asymmetry. They keep phosphatidylserine strictly confined to the inner leaflet. During cell death, this exposure acts as an eat-me signal.
In cells undergoing necrosis, a massive influx of calcium activates the scramblase TMM16F, exposing PS before membrane rupture. During apoptosis, caspases cleave flipases. This activates the scramblase XKR8, leading to rapid phospholipid scrambling.
Once exposed, PS is recognized by phagocytes via direct bridging molecules like Gas6 bind receptors. In the brain, MEGF10 functions in astrocytes to clear debris.
Apoptotic cell uptake is superior. They are internalized as condensed particles, whereas necrotic cells take up as fragmented pieces. Neither process induces inflammation. Apoptosis is programmed and caspated. Secondary necrosis occurs when a clearance fails. Primary necrosis is unregulated, necroptogrammed but caspase-independent.
Apoptosis triggers excate, flipping lipids. Without clearance, degrades the membrane. Accidental necrosis floods calcium. Necroptosis executes a programmed lytic rupture.
Apoptosis is rapid and highly efficient. Secondary necrosis debris is cleared much slower. Primary necrosis requires complete disintegration. Local phagocytes engulf necroptotic debris. Tissue-resident macrophages recycle iron. They derive this iron from senescent red blood cells. This process is erythrophagocytosis. It sustains hemoglobin synthesis. Aged erythrites undergo membrane modifications. Scavenger receptors on macrophages initiate phagocytosis. The RBC-containing phagosome matures. Fusion with iron.
Phe2+ enters the macrophage pool. It is stored or exported by ferroportin. Ceruloplasmin oxidizes it. It binds transferrin for transport to the bone marrow.
FPN expression is tightly regulated. Erythrocyte iron directly induces its mRNA. Levels increase 8-fold within 4 hours, while FPN protein peaks at 10 hours. This is blocked by chelators. Hepcidin binds to ferroportin. This triggers internalization. Ubiquitination leads to endocytosis. Lysosomal proteolysis degrades the complex. Inflammation induces this pathway. Systemic iron levels regulate hepcidin. Circulating iron binds to transferrin receptors. This activates signaling through BMP receptors. This drives hepcidin gene transcription. Erythroblasts secrete the hormone erythroferrone. This suppresses hepcidin synthesis in the liver. To maximize essential iron export,
Low hepcidin stabilizes ferroportin on macrophages. This activates SYK and downstream calcium signaling, expansion of cellular networks. Cellular remodeling is fueled by a metabolic shift. This fuels the enhanced clearance capacity. Pharmacological inhibition reverses this adaptation. This program is unique to splenic RPMs. The primary pathway governing this self-recognition is the CD47 alpha axis. This checkpoint prevents autoimmunological attacks on healthy tissue.
CD47 is a marker of self on host cells. It binds to SIFA on macrophages. This recruits SHP1 and SH2. This action inhibits macrophage attack.
During activation, opsonized targets bind to receptors. This recruits Syk kinase. This drives rapid F-actin rearrangement. Lamellipodia engulf the target.
CRP alpha is engaged by CD47. Recruited SHP-HP2 phosphatases directly dephosphorylate VAV1, blocking RAC activation and halting phagocytosis. Consequently, opsonized targets expressing functional CD47 can only be internalized via a less efficient, slower, Rho-mediated sinking mechanism. Macrophages pig-to-human grafts face rapid rejection. Donor CD47 fails to bind human CRP alpha. Transgenic pigs now express human CD47. This effectively prevents graft clearance. Vesicles overexpressing CD47 can transfer this protein. This cross-dressed CD47 interacts with S-alpha to inhibit phagocytosis, protecting the transplant. When foreign dead cells enter a host, they release universal DAMPs like ATP. These molecules recruit host phagocytes by activating conserved GPCRs. Phagocytes arrive at the focus. But clearance deviates due to the lack of self-signaling. Foreign xenoantigens like alpha-gal trigger rejection. Humans possess natural anti-gal antibodies. They bind and initiate complement activation. Complement opsonins deposit on dead cells. Anaphylotoxins recruit more immune cells. Host phagocytes engage via Fs triggers robust Syk-dependent signaling, creating an inflammatory environment. Because there is no functional CD47 SHP phosphatases and dephosphorate and dephosphorylate VAV1, the host macrophages treat the foreign DES as active pathogens.
At the interaction node, human CD47 binds to CYRP-alpha. This high affinity 2 inhibiting RAC-mediated reaching. This preserves the healthy self-cell.
Interaction node. Binding affinity is low, at 6.93 micromolar. This drives unchecked Cyc-RAC activation, resulting in rapid, reaching phagocytosis.
Between human and porcine cells, CYP-alpha binding is negligible. This triggers anti-Gal binding and classical complement activation, resulting in lysis and inflammatory macrophage activation.
At the interaction node, CD47 binds to CYRP-alpha. CD47 phagocytosis, promoting graft survival without transmitting cell death signals. At the interaction node, binding affinity remains unchanged, yet downstream effector cascades alter the fate, clinical inhibition, and accelerates clearance of rigid cells.
Laser-induced injury models have advanced the study of necrotaxis. Researchers can target sub-micrometer regions. This shows leukocyte recruitment in real time.
By titrating the laser input power, we induce specific DNA lesions, from simple single-strand breaks to complex double-strand breaks. Confocal microscopy suffers from two major limitations: significant light scattering and severe phototoxicity. TPLSM overcomes these limitations.
Immediately following damp gradient exposure, migrating leukocytes undergo an initial search phase, changing direction as they orient along the gradient.
Once oriented, the leukocyte enters the run phase. This phase features rapid, polarized actin flows. It drives directly toward the necrotic focus, sustaining high directional persistence.
By combining high-resolution imaging with genetic and pharmacological perturbations, researchers can dissect the fundamental rules that govern leukocyte behavior. Researchers use laser ablation to create precise, sterile wounds. This triggers immune cell migration to monitor leukocyte recruitment.
Like what you shares.
This website makes learning so easy. I found all my resources here.
Noir plasma chrome bleed. Unlocking knowledge, inspiring innovation, empowering curious mind.